Hello, everyone. For the next 25 minutes, I'm going to tell you a little bit about artificial intelligence and deep learning and how these methods can accelerate Alzheimer's disease research. And I'm going to tell you about work by a consortium known as AI for AD, which is a newly funded grant from the National Institute of Health. So just a couple of disclosures. I received partial grant support from Biogen and Kairos Venture Capital on applications of AI. We'd also like to acknowledge the funding from the NIH and the Alzheimer's Association in supporting these machine learning projects. So a quick summary of the talk is as follows. We're all interested in how we can use artificial intelligence in medical imaging, and I'll talk about a few example applications from Alzheimer's disease research, using machine learning and AI for diagnosis and prognosis and subtyping of disease, inferring neuropathology from different types of medical images, and also to help us disentangle disease processes in population studies and for drug trials. Now, the flip side of this is that there are a few pitfalls and common issues with applying machine learning to multi-site data, and I'll tell you a little bit about those as well. So the first is how to deal with domain shift when you train and learn a predictive model from a certain data set, but then you go on to apply it to another data set and it might not perform quite so well. Another approach using deep learning is to use image harmonization, where you adjust new sets of images to match the data that you trained your machine learning algorithm on, and I'll tell you a little bit about that. And then also some new opportunities to combine neuroimaging and genetic data to discover genetic markers associated with accelerated brain aging and disease. And this will involve applying AI methods to whole genome sequences as well as medical images. So when we talk about AI and deep learning, particularly applied to a disease such as Alzheimer's, we're interested in taking all of the aggregated biomarker data from patients, whether it be genome sequences, omics data, blood tests, neuroimaging, and even real-time data from activity monitors such as an actigraphy watch, and essentially building a predictive model from this that might help us do tasks such as diagnosis, prognosis, infer likely treatment response. And AI methods are very useful in discovering and learning patterns in biobanks, huge amounts of data from patient populations. And we can train and use these methods for diagnosis and also subtyping. So in Alzheimer's disease research in particular, we're very interested in understanding the mixture of pathologies that are going on, whether it be amyloid or tau or neurodegeneration or even vascular disease. And we're also interested in using these methods for predicting future prognosis and treatment outcomes. So common deep learning methods include a variety of neural networks. Convolutional neural networks are very appropriate for analyzing medical images. Also generative adversarial networks, I'll tell you a little bit about, also for working with images. And also for working with genome sequences, recurrent neural networks, RNNs and their variants have become very popular. All of these methods are used to discover patterns that are associated with a certain outcome or a certain decision. So in the network at the top there, we might be developing a biometric authentication system to detect whether a person should be let in to a facility. This is a picture of someone called Sarah. And a neural network might learn to recognize her essentially by distilling higher and higher order features from the images. If there are a number of images, we'll train a classifier to say whether this person is her or not, and these types of methods work very well. In a medical context, we often are taking a patient's MRI scan or other type of medical image, and we want to make assertions about their clinical diagnosis, what types of pathology they might have. And in benchmarking tasks, just to test these algorithms, we often try and predict the person's age, and I'll tell you a little bit about that problem also. So this has been immensely successful. A really fantastic paper, remarkable work by Bin Liu and Xiaogan Yan at the Chinese Academy of Sciences. They trained an Alzheimer's disease classifier on MRI scans from, in fact, 50,000 people or 85,000 scans from 218 sites and scanners. And what they did is they pre-trained the network on a sex classification task, trying to tell if the MRI was from a person who was male or female. And once the network was good at that task, they fine-tuned it using transfer learning to classify Alzheimer's disease. And they were able to do that across all those data sets with a little over 91% accuracy. Now, even beyond diagnosis, we can infer from images information that we may not have collected or may not even be able to collect. So Duigu Tosun, who's a member of our AI for AD initiative, and she's a faculty member at UCSF, she has trained a multi-label random forest classifier to infer whether there are different types of neuropathology in the brain based on paired autopsy data and pre-mortem imaging data. Now, obviously, we don't always have autopsy data from patients, but it's of interest to know if the cellular pathologies such as TDP-43 or Lewy body dementia or cerebral amyloid angiopathy are present. And this type of disease subtyping might help us to identify patients that might respond better to certain types of treatments, for example, treatments for vascular disease, and also to stratify drug trials or even genetic or epidemiological studies by better defining the biology that's going on in the patients that we're looking at. Now, deep learning wasn't always popular. So my first graduate student, Alain Pitio, was developing a deep learning method to identify the corpus callosum on MRI using really a very early deep learning neural classifier. And a lot of our collaborators at the time at the INRIA medical research group in France said that they didn't really prefer to use artificial neural networks. They said, well, how can we see how it works? There's no simple analytic model or variational formulation or partial differential equation to help us understand how to evolve features in these images and detect them. And so this was not entirely popular at the time. And so this has led to an interest in interpretable deep learning. So many of the methods that we apply, and I'll show you today, not only make a decision based on a scan, but they give you a heat map or a saliency map of all of the features that we used. And they may include features that we didn't anticipate would be useful for making these decisions. So both saliency maps and also deep learning networks using attention mechanisms that focus on particular regions of the images are becoming very valuable indeed. Work by Andy Sakin and Tai-Ho Zhou at Indiana University is using tau-sensitive PET to feed into a deep learning neural network, again, a convolutional neural network, to identify features and sets of features that are diagnostic for Alzheimer's disease classification. And this can often identify things that we didn't, again, expect ahead of time that would be diagnostic or prognostic from PET. Now, it's a bit of a simplification to say that disease processes evolve identically in all patients. And so another use of machine learning, very elegant paper by Jacob Vogel and colleagues here, is to do subtyping and staging. And so you may have collected a series of longitudinal images from a large number of patients. And it could be the case that you could cluster those spatiotemporal evolution patterns into distinct trajectories that characterize different subtypes. And so they found that even within tau PET, you could not only identify different patterns of progression, but also assign a score to each patient for each subtype to help us understand what type of disease processes were going on, and also to stratify information if we wanted to go ahead and do a treatment study or a genetic study in these more biologically precise categories. Now, there are also some pitfalls to watch out for when applying AI to medical images, and I'll tell you about a few of these. The first challenge applying AI to medical images is domain shift. So you could have a predictive model that works extremely well when applied to images from a certain scanner, the scanner it was trained on, but it might then fail badly when it's applied to images from another scanner or another site. Now, some of the solutions proposed for this are to train and test algorithms on more diverse data, but also to use transfer learning, where we learn a deep learning model from a certain data set, and then we fine tune it to be more accurate on a new data set that we might be interested in making decisions about. An even more difficult problem is to avoid confounds and to avoid learning site-specific effects. So I'll give you an example of this. If you train a deep learning model to make a prediction, it can lock onto a confounding feature at a site. So let's say patients at a certain site have a more severe disease, more atrophy. The AI method may think that all the images from that scanner indicate more severe disease. Or another example is, maybe one site scanning protocol depletes signal a little bit in the frontal lobes, and the AI might think that that's a new subtype of dementia. In fact, it's just a technical artifact, but it might think that that is a new type of disease. So I'll tell you a little bit about how to mitigate these. There's some really nice review articles and technical methods that have been developed to address these. First of all, for avoiding biased conclusions about the accuracy of data sets when confounds are present in the data. And then also in the second paper here, very nice recent paper, preventing data set shift from breaking machine learning biomarkers. They're talking about this domain shift issue where you train on a handful of data sets and then your methods break down on the data set that you really care about. So one of the ways that we control for confounders in normal statistical modeling is we regress them out. But as this paper by Richard Dinger mentioned, it's extremely difficult to remove all the confounding effects from a data set of images. And it's really insufficient because deep learning methods can also still find features in that data that might be associated with a confound, even though you think that you've regressed them out. So one of the approaches that has been developed to develop predictive models with deep learning that are robust to confounding effects is to notice that if a hidden confound influences both the input and output of a machine learning method, the machine learning method may lock onto it and find a spurious association. So what Qingyu Zhao and Kilian Pohl advised to do is to learn features that not only successfully predict an outcome, it might be Alzheimer's diagnosis, but also use an adversarial subnetwork in the deep learning approach that tries to predict the confounder. So we've already mentioned that the confounder might be the site where the patient was scanned, or it could be their age or sex. It could be features that are not relevant to the diagnosis and can get in the way. And this type of adversarial learning approach can iteratively adapt the feature set to make it impossible for the adversary to predict the confound from the features. In other words, it's eliminated information about the confound from the features. And one very elegant application of this was proposed by Dan Moyer, who was a former student of ours at USC. So he said, can we use a variational autoencoder, which is a type of data compression technique, in conjunction with a generative adversarial network, where we can adjust data coming from different sites and scanners to look like the images had been scanned on a certain scanner. And if we did that, the domain shift, the change of venue for applying a deep learning algorithm might be mitigated to some degree. So let's see if we can tackle this question. How would your brain look if you'd been scanned on a different scanner, or for that matter, with a different protocol? So what he does is he uses a neural network with two components. One is a variational autoencoder that takes the initial scan, it might be an MRI scan, and essentially embeds all of the features in that scan into a latent space. And that latent space is going to be used to make predictions, diagnostic predictions, and so forth. At the same time, we're going to have an adversary, a specialized neural network that's designed to tell where the images come from, which site they come from, and which protocol was used. And what he found is that you can train a neural network that maximally predicts a certain outcome. It might be Alzheimer's diagnosis, but makes it difficult or impossible for another neural network to tell where the data came from. And in doing so, it gives a site-invariant set of features that not only are useful for prediction, but also can be used to generate images with a different contrast. So what he essentially does is he takes MRI scans from many, many different sites, saves only the biologically interesting information in those scans, and eliminates the characteristics that have to do with the scanning protocol or the scanner, and then uses the siteless data essentially to make predictions. And you can also recombine this with information about the site to generate data that looks like it had been scanned on a certain scanner. So this eliminates the site effect or the confound when training and testing machine learning algorithms. A very elegant explanation of this was given by Aisha Dinser. She's a researcher who works on gene expression data. And she said, you know, essentially when we're training machine learning algorithms to find patterns, we have all sorts of different sites and sources the data are coming from. And we don't want to be subject to this domain shift where data from a new site cannot be classified or cannot be used because it's somehow too different from the data that was used for training. So what she does is she essentially takes all of the data that's used for training and testing, again uses a variational autoencoder to embed the most important features for a certain task, and also uses an adversarial network that tries to tell where this data came from. And you can imagine a massive numerical optimization problem where you try and find features that are not only informative about a certain task, but also make it impossible to say where the data originated. And these formulations here can be used to train all of the hidden layers in the network and make, in a sense, bias-free predictions. And these are implemented with very special layers in neural networks, one of them called a gradient reversal layer, that allows you to provide feedback on confounds into the main training process for a primary task. Now, this type of adversarial harmonization is very, very useful in imaging. So if, for example, in this work by Mengting Liu in Neda Jahanshad's lab at USC, you might have collected data at all sorts of different sites, trained a machine learning algorithm on some of them, but want it to perform well on data from another site. These are different projects like ADNI and UK Biobank and so forth. And what's happened here is that the images there at the top are being used as a reference, and we're trying to convert all of these different scans so that the neuroanatomy remains the same, but they exhibit the contrast and so-called style of the scanner that is specified at the top there. And this allows direct harmonization using this generative adversarial network of images from very diverse sites and scanners to really have the same contrast and not then fool or make machine learning methods trained on just one site break down. Now you can take this a little bit further. The Hopkins group, led by Jerry Prince and Aaron Karras here, have developed a way where you do adversarial correction of images from different sites. In other words, you can make an image from a certain site look like it was collected elsewhere. But you also learn a latent subspace that allows you to have controllable contrast. And so on the left there, images from different sites, there are 8 of them in this example, their contrast has been essentially mapped to a certain underlying parameter space where you can controllably vary what type of contrast you want and have these images match a contrast, a desired contrast from a reference data set, thereby improving the results of disease classification and prognosis and eliminating this domain shift. Now, this is a very, very popular approach. It can be extended a great deal. This work by Nicola Dinsdale and Anna Nambarete at Oxford University is looking at how we would make predictive models that adjust for other confounds, not just the site where the data was collected, but other confounds might be age or sex, or there might be something about the patients being scanned. And so what you can then do is embed adversarial networks that make all of the predictors used invariant to the confounds in the study, and essentially unlearn those features by adjusting the feature representation to make it impossible to predict the confounds. And then you'll end up with a predictor set that you're using for machine learning that is absolutely robust to where the scans were collected, whether the person it's applied to is female or male, or how old they are. And these gigantic simultaneous optimization models are really being explored right now for making MRI-based AI more robust. Now, does this work? And it does. So this work by Surbhi Sinha, working with our lab at USC, she applied GAN-based correction of MRI scans and showed that it really doing this, in this case using a CycleGAN approach, can greatly improve disease classification. So instead of applying them straight out of the box to scans from a new scanner, she applies this generative adversarial network to correct the scan. And then the performance, in this case of classifying whether someone has Alzheimer's disease, goes up by about 7 or 8 percentage points in terms of balanced accuracy. So these have been very powerful and useful in MRI research. Now, in the final 5 minutes, I'll tell you a little bit about using neuroimaging data to advance our understanding of Alzheimer's disease genetics, which is a little bit of a merger of 2 fields, to use MRI scans as a target for genetic analysis. I'll tell you about some standard methods for imaging genetics that are used in our Enigma consortium, and then also how people are beginning to extend these methods to novel AI and machine learning approaches to discover factors in the genome that are predictive of outcomes and features in images. So overall, we might want to discover genetic markers that are influencing the brain or brain disease. And the brain images themselves might be a good target for genetic analysis. So often we want to associate variations in our DNA genetic sequence with a disease. This often requires 50 to 100,000 people to identify robust variants in the genome that are associated with a disease. But we could also do this with images. We could find genetic variants that are associated with a certain pathology that's evident on imaging. And that might be interesting to develop drug targets and also to stratify patients in epidemiological studies. So what you do when you're doing this is you march through the genome, and at each locus or single nucleotide polymorphism, you run a regression. You ask whether that marker in the genome is associated statistically in a population with some measure from the images. It could be the size of the brain, or it could be the PET signal in the temporal lobe, or something else. And then you identify the markers in the genome that are predictive of the measures in the image. And when we've applied this in our Enigma consortium across the globe, over 2,000 members are participating, and now over 100,000 MRI data sets, you can really develop tremendous statistical power to identify variants that matter for the progression of disease. So all of these sites in Enigma might compute brain measures from their scans, morphometry measures from MRI, or white matter microstructure measures from DTI. They'll run a genome-wide association to identify significant associations between the brain measures and the genetic variants or the SNPs. And then in a meta-analysis, you can combine all the evidence across the sites and each site vote can be made to depend on the sample size it has, so that it makes sure effects are reproducible and also boosts the power to pick up effects that the single sites could not identify on their own. This is very useful. So in a project here that pulled data from the CHARGE Consortium, Enigma, and the UK Biobank, the question was asked about white matter hyperintensities. In other words, were there any variants in our genetic code? If you see the plot on the left, that's our chromosomes on the x-axis, and the peak there is a hotspot of association between a genetic variant and the volume of these white matter hyperintensities. And the question asked here was whether distinct patterns of vascular disease were associated with different genetic variants, and there's a little bit of evidence for that. This could also be applied to measure any part of the brain structure or function in an image. If you travel around the cortex now, a little over 200 genetic loci have been discovered that influence the thickness and surface area of these different parts of the brain. A little over 40 loci have been discovered that affect the volumes of subcortical structures. And a lot of interesting science has come of this. So risk genes for Alzheimer's disease include the ApoE4 haplotype, which around a quarter of us carry. And it triples our risk for Alzheimer's disease for each copy of ApoE4 that we have. And in these imaging genetic studies where, in this case, hippocampal volume was measured from scans across 60 worldwide sites, we were able to notice that this ApoE4 genotype is associated with, in fact, a 2 standard deviation reduction in the volume of the hippocampus, helping to explain why it is a risk gene for Alzheimer's disease. It seems to exert its effect directly by affecting hippocampal volume in this study. We also can explain about 4 to 5% of the variation in hippocampal volume on MRI by compiling all of these variants in the genome that have been discovered by genome-wide screening of scans across the world. And now over 500 loci have been discovered that affect subcortical volumes, over 50 for hippocampal volume alone. And these can be very interesting to test. You can compile these genetic markers and see how well we can predict, in this case, hippocampal volume from genetic loci. And you can explain around 4 to 5% of the variance in these volumes. It's very, very useful to do this. We've also been looking at sequential MRI, people who've been scanned twice with MRI, and you can create an estimate of the rate of atrophy of different structures in aging and their rate of growth and development. And we've discovered, this is work by Rachel Brower and Hilke Hulshoff-Paul, over 15 loci in the genome beyond the famous Alzheimer's risk gene ApoE that affect the speed of brain aging. Very, very interesting to mine the genome using images and discover markers that could be dangerous and then could be a potential target for drug development in interfering with the cascade that arises from having these variants. We also know that mass univariate detection of genetic features is not optimal. And so here's work by Marco Lorenzi at INRIA in France, and also work by Yunga Chen and Li Shen in our AI for AD project, where they've asked the following question. They've said, can you tell me what combination of markers in the genome is most predictive about the signals all over an image? In other words, this could be set up as a canonical covariates analysis problem. Could you find the optimal linear combination of markers in the genome that explains the most about the images? And we're also allowed to use a weight map in the image, which is also a linear combination, and essentially optimize over both data sets at once to, in many cases, find an optimal, sparse, and efficient set of explanatory variables. There's a lot of interest in discovering influential genetic loci that also lie in druggable pathways. In other words, we have pharmaceutical treatments that can influence their activity. So in a lot of this work, not just deep learning being applied to genomic and imaging data, but also bioinformatics data is being brought in to guide the selection of genetic markers that might actually be among those that we could repurpose drugs to influence. And so there's a lot of cooperative work between bioinformatics and, in a sense, unsupervised deep learning so that the two approaches can benefit from each other. So just to wrap up, some recent successes of artificial intelligence and machine learning in Alzheimer's research include novel automatic methods for diagnosis, prognosis, and also subtyping and staging of disease. So not only saying whether a patient has a certain disease based on their imaging, but also saying at what stage they're at, whether there's a mixture of pathologies, and also, as we saw in the neuropathology example, what are the cellular correlates likely to be. Now, these patterns are often learned from biobanks of MRIs, PET scans, and even neuropathology. And so some hot topics, really exciting areas include pathology detection from in vivo imaging, where you have autopsy data also to help train the methods. Again, dementia staging and subtyping is very important. And also data harmonization using AI. We talked about how generative adversarial networks and variation autoencoders are easing the domain shift that's inherent in applying deep learning to new data sets by either adjusting the model to adapt it to the features of new data, or adjusting the data so it resembles the data that was used for training algorithms. We're very interested in some open questions. Can we use AI and machine learning to relate dementia subtypes to genetic subtypes? So that method known as deep sparse CCA might find a combination of motifs or features in genome sequences that are related to the dementia subtypes. It'd be very interesting. And also whether we can jointly analyze whole genome sequences, the 3 billion letters of our DNA and brain imaging, to unearth some predictors that affect, in the example we showed, the speed of brain aging, or that might affect treatment response. And this is a very, very active area of work, and new initiatives such as AI for AD are beginning to use not just deep learning on its own, but also guidance from neuroinformatics databases, particularly something called the Placebo system, which Gunga Chun has developed at Boston University, to combine knowledge on specific druggable pathways with discovery methods in AI that pull out of the genome potential targets that are affecting disease onset and progression. So just to wrap up, thanks so much for your attention. And we're very appreciative to all the AI for AD members and also the Enigma members that contributed to the work presented here, and also to the NIH who are funding this project, and the Alzheimer's Association that have contributed funding to many of the efforts that I've described today. So appreciate your time, and thanks so much for listening. Bye-bye for now.